بسم الله الرحمن الرحيم. السلام عليكم ورحمة الله وبركاته. أهلا وسهلا فيكم في المحاضرة السادسة، وهي المحاضرة الأخيرة في مساق المقدمة إلى علم البايوانفورماتكس، أو ال- 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 المسمى الرسمي للمساق هو Selected Topics in Artificial Intelligence. طيب، الحمد لله على سلامتكم كمان مرة. أتمنى إن شاء الله تكونوا بألف خير وربنا يحميكم يحفظكم. العالم رايح لناحية تانية، إن شاء الله ربنا يسلمكم جميعا، وبتمنى ربنا يعم السلام والخير على منطقتنا ويحميكم، وتنتهي الحرب إن شاء الله قريبا بأقل الخسائر، ونشوفكم على خير قريب. طيب، تكلمنا خلال المساق في عدة مواضيع، من ضمنها كنا بنتناقش كيف السيكونسينج ماشين بيعملنا generation لهيوج data. الداتا هاي has no information at all where is the exact source of each piece. يعني ما بنعرف السورس تاع كل قطعة بناخدها من السيكونسينج ماشين، هل هي جاية من كروموزوم 1 كروموزوم 2 أو من أي جزء من الجينوم تبعنا. وبالتالي كنا نعمل شغلة اللي هي زي البازل، ناخد reference picture، نحاول ناخد هاي البيسز ونقارنها بالreference picture، ونحاول نعمل attach together, so we rebuild again the whole puzzle, or we solve it so that we could get the full picture of our genome. Okay, and we talk about a difficult puzzle that when we don't know the reference genome, or we had two samples from the same individual, so we'd like to map them to each other to identify differences between two FASTQ files. Another very difficult puzzle when we really don't know the reference genome, or when we don't know what we are sequencing. So you just collect a sample from environment, could be train seats, bus seats, university desks, and so on, where you expect to have many bacteria, viruses, or diversity of those collection, basically. So when we don't know the reference genome, we cannot compare those pieces coming from the sequencing machine to a single reference genome. What we do is we bring a database of all known reference genome that we have, and then we start picking reference by reference. So we compare those pieces to all those references and try to make best guess of which reference genome share large similarity with the pieces that we have coming from the sequencing machine. In this course, we focus on the first scenario where we solve the puzzle itself, assuming that we know the reference genome and assuming that we have those pieces from single sample, and we use sequencing machine to generate those pieces. And let's do a formal definition of read mapping. So read mapping is a computational step، وبالتالي هي خطوة حسابية أو عملية حسابية أو عملية تحتاج إلى خوارزمية أولا، ومن ثم implementation في لغة برمجة، ومن ثم تطبيق أو execution تنفيذها على جهاز الكمبيوتر. بالتالي هي computational step that locates، يعني هي اللي تحسب وين مكان كل قطعة أخذناها من sequencing machine. possible subsequences of the reference genome. ليش subsequences مش subsequence؟ لأنه غالبا لما ندور على كل قطعة جاية من السيكونسينج ماشين احتمال يكون هناك تشابهات عدة في الريفرنس جينوم، وبالتالي لما نقارن مع الريفرنس جينوم ما بنقارن مع قطعة واحدة، أو ما بنتوقع أن نشوف التشابهات مع قطعة واحدة من الريفرنس جينوم. وبالتالي we are trying to locate possible subsequences of the reference genome that are similar or share similarity to a read sequence while allowing at most e-edits. So why we are allowing for those e-edits? The E is edit distance threshold here. And it's very important to allow for those differences, because when we compare our own pieces coming from the reference genome to the coming from the sample that we sequenced, and when we compare it to a reference genome, this reference genome belongs to someone else, right? So in order to maintain correctness So when we compare our pieces with that reference genome, we should assume that we will have some differences by nature, right? And those differences includes the genomic variations. So we know that each genome is somehow different from someone else's genome. There are some similarities, right? Because all of us are human, but there are also differences, which makes us different, right? Including skin color, eye color, hair color, and many other traits. Some of the other differences when we do the comparison is a result basically of the sequencing machine, because sequencing machine is still a machine, right? They are not perfect. They introduce their own error, and those errors normally depend on the sequencing technology that we use. So we agree that there are now 3 major technologies: Illumina, Nanopore, and HiFi, coming from PacBio. So each of those sequencing technology introduced their own error. Some of those, like Illumina, for example, introduce errors toward the end of the read. So the last 30 bases of each read normally are errorless, right? And some other sequencing technology introduce different kind of sequencing errors, including insertions or deletions. So some of the sequencing technology are very famous for only insertions. So they introduce their own insertions. So it's very important to maintain the correctness of read mapping by tolerating some differences when we do the comparison. That's why we cannot do exact match all the time. So we cannot expect to have the exact same piece that we get from the sequencing machine to exist in the reference gene. So this is a very important aspect of read mapping. And we already have seen this slide before in one of the lecture in this course about a brute force algorithm on how we do read mapping, right? So imagine we have a reference genome and we have a read, and we would like to scan every single piece or every single location in the reference genome and try to check whether it's similar to the read we have or not. However, we agree that this is very expensive because we are using dynamic programming algorithm, which takes quadratic time calculation in order to check the similarity between 2 subsequences and see how many differences are there optimally, of course. We could use some pre-alignment filtering mechanism to approximate that number, but normally we look for an exact solution, the accurate number of edit distance, for example. All right, so we also went through this practically on how we perform read mapping. So we agree that we need a FASTA file that represents the reference genome, but we also need a FASTQ file that represents the sequencing data. And we collect some information from the FASTA file, and that step we call it indexing. We already give you examples and quiz questions. Hopefully this is very clear on how we pick those seeds. Of course, we give you an example of 2 mechanisms called genome on diet and minimizers, but there are many other ways on how we can collect those seeds. You could also do the exact same thing to the FASTQ file. So we collect those seeds, but we don't index them. We use them to query what we built in the FASTA file step. And then we try to locate where exactly in each reference genome or at each location in the reference genome that a subsequence shares some similarity with what we have in the FASTQ file. And we said because we have 3 seeds appearing next to each other, blue followed by green followed by red, so we expect to have them in the exact same way in the FASTA file. So we are looking for blue followed by green followed by red, which is somehow this portion of the reference genome that shares a similarity with the FASTQ file. And then we try to find the mapping location. So we have a reference genome, we have also a read. What we do, as we said, we collect some seeds, somehow assume the method that you like, and the next slide we will see some examples of those. And then when you collect those seeds, you record every single seed in a hash table, for example. Hash table is just a mechanism of indexing, but there are other methods that you could use to record those seeds and query them quickly later on. So if you use a hash table, what we need to store is basically the seed itself, which normally we store it as a hash value, not the content, but we store it as a hash value. And then we store a list of start location of where each of those seeds appears as a first character in the reference genome. So we have the green seeds over here, assume the length, for example, 16 character in this chunk, right? And what we record over here is the location of the first character in that chunk, whatever it is, 0, 1, 2, 3. And then we build the index with that. And you already have some examples on how we collect minimizer seeds and how the hash table would look like out of those seeds that we collect. And this is the example, one of the examples that we have. So we are trying to locate this read in the reference genome, at which location we would see a subsequence that is similar to this read sequence. So we have a reference genome, which is usually much longer than the read sequence itself. And what we try to do, we are trying to get the location or try to locate where this read sequence appears in the reference genome. Again, we're not looking for exact match, but we are looking for approximate exact match or approximate matching. So what we would do, we would get the reference genome first, right? And then we collect the seeds, depends on the way that we would like to use or what algorithm that we are going to use. Here we are using a minimizer algorithm with k equal 10 and w equal 5. We are also using lexicographical order to as a mechanism to choose which seed out of the w seeds we should choose. And those are the minimizer seeds that we collect out of the all overlapping seeds that we collect from the reference genome. And then we choose some of them to be stored in the hash table, right? And then with that, this is our hash table. This is the thing that we can query to quickly check if a subsequence of the read exists and how many of those seeds exist in the reference genome. This is just a brief overview of the methods or the algorithm that you could use to extract the seeds and to index them, basically. So we have overlapping method, which is all the previous seeds that you have seen here. So you could choose all of them to be included in the hash table, or you could choose non-overlapping. So you just skip those that are covered in the previous seeds. For example, you pick this seed, and then you move on to this seed directly, which is non-overlapping with that first seed. But you also have spaced where you could skip some of the characters. So this is the first seed, you could skip the second character, you move to the second seed, you skip the second character in the second seed, and so on. And with that, we call it space seed because we make a space in the k-mer itself. And the benefits of doing so is allowing for mismatches at the second position, for example, because we don't require that character to exist between the two seeds to match them. We also have adjacent, non-adjacent, syncmers, dropmers, genome diet, minimizers, and compressed seeds. And the performance, those methods or the choice that we have for choosing those methods over the others will directly impact the performance of the tool that we are building to do read mapping. So you can see there is a drastic difference between the tools that are doing indexing, and you can see we could go from 3 GB all the way to 17 GB. And the indexing times can also be very huge because the computation or the workload that we are doing at each of those algorithms is totally different. It can go from 3 minutes all the way to around 49 minutes, or from 1 minute to 49 minutes or so to just build the index. This is the performance that we did in this paper back in 2021 about various methods, around 108 tools that we evaluate and survey in the paper. You can see the performance. So in this plot, you can see the CPU time of indexing alone, just indexing. If you see the hashing, some of the tools are very fast, below the average, right? And some of the tools are extremely slow. They are much slower than all the other methods. When we look for Borzweiler transformation, FM index, in those tools, normally the execution time is very close to each other because we are doing here compressed indexing. So the seeds themselves are compressed. So by saying compressed means it's a tree structure. So if we have some of the path shared between two seeds, like the first 5 characters are common, we don't record those seeds separately, but we merge the first 5 characters together, and then we have the rest of the seeds split it over different paths. We have other methods for indexing, like suffix tree and so on. And when we look into the memory in gigabyte, this is the peak memory of the indexing step, how much main memory we are occupying. So when we look to the hashing, it's also same story as the CPU time. Some of them show very small memory footprint, and the others show very large memory footprint, even much larger than what we have from the other techniques. Again, what makes a tool slower or faster or having large memory requirement is the method that we are requiring. If you are going to consider all overlapping seeds, then the number of seeds that you are going to store in the hash table is tremendous. And that makes the indexing step requiring a huge main memory. It's very slow because we are going to calculate every single seed and go to the hash table, try to search, does it exist? If it exists, what location? If the location is unique, then we record it. If it exists already before, then we ignore it, and so on. So there are a lot of calculation happening between the main memory, the caches, and the CPU. Now, after we build the indexing, what we need to do is to do the exact same step, which we call it seeding. In seeding, we don't build index, but we use the exact same algorithm to collect the seeds from the read, and then we use those seeds to query the hash table that we built in step 1. So in seeding step, we got the read sequence. We use the exact same method you use in reference genome, whether it's minimizer, genome and diet, overlapping, non-overlapping, or any of those methods. The exact same way you use it for hash table, for reference genome, we should use it in read sequences. And then we pick seed by seed and we check, does it exist in the hash table we have? If it exists, at which locations? So we got this list of location as a result of the query. Once we get the list of the location, we try to match those location all together. So from seed, the first seed we query, we got all those lists of locations. When we got the second seed, we got all those lists of locations. When we query the third seed, we got those lists of location, and so on. And then when we know that those 3 exist in one of the read sequences, we try to sort all those locations to make sense out of them. Because we already see in the previous example that we wanted the seeds to be in the exact same order as they appeared in the read. So if they appeared in the read as yellow, followed by blue, followed by green, then we want them to appear exactly the same way in the reference genome too. So when we sort the location, we will see that we have 5 as a location for the seed, for the yellow seed, and then we have a 7, we have a 9. It means that all those seeds are respecting the order that we have in the read, which makes this particular subsequence in the reference genome somehow similar to the read sequence we have. However, what we do now with the seeding is that we extract the seeds which could be apart from each other. So there is a gap between every seeds. في عندنا فراغ between التو seeds، وبالتالي هذا الفراغ معناته انه ما فحصناه حاليا، يعني ما فحصناه، اللي فحصناه هو exact match between the seeds. يعني تأكدنا انه المربع الاخضر والمربع الاخضر موجودين بين الريد والريفرنس جينوم، نفس الشيء الازرق والاصفر. وبالتالي we never yet check the characters between the seeds, whether they also match with what we have between the two seeds in the reference genome or not. That's why we need the next step. So now let's see an example on how we do seeding to the read. So we already got some examples on the reference genome. Now let's get the read that we have it a few slides before when we try to locate this read in the reference genome. So this is the read sequence, and the question asks us to use the exact same method that we used in the reference genome. So we try to extract all the overlapping seeds, and then we consider every W of them. W here is 5, but we have only 4 seeds could be extracted from this read sequence. So we consider all of them basically. And then we try to see which one is lexicographically minimum to save it or pick it as minimizer seed. And we find the first one is the wanted Kamer. However, remember, we don't want the location, so we don't record the location in here. And when we do seeding, we don't need the location of the seed in the read itself. What we do, we just have that seed, and then we could use it to query the hash table. Does it exist in the index or the hash table? Yes. At which location? It seems it exists at location 0 and location 6. Now, since we never checked the characters in between the read sequence and the reference genome, now it's the time to do thorough analysis or thorough examining for the two sequences to see whether they match fully, or if not, there are some differences. How many of those differences? Does it exceed the edit distance threshold or not? Now in step 3, when we want to do the pre-alignment filtering to check the entire similarity between the read and the reference subsequence, and now based on querying the hash table, when we do the seeding, we know that this seed exists at location 0 and at location 6. So we go to that location 0 at the reference genome and try to pick a number of characters that is equivalent to the read sequence that we have. So again, because we use the minimizer seeds that we extract from the read sequence to query the hash table, and then we know based on the similarity or the matching between the seeds that this seed matches the reference genome at location 0. So we go to location 0 in the reference genome and we pick the exact number of characters that we have in the read sequence. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So we pick also 13 characters from here, and then we apply a pre-alignment filtering algorithm on that. So we have the read, the reference genome. We try to build here, for example, Sneaky Snake, which is the maze to calculate Sneaky Snake algorithm. And then we build this based on those two sequences and check what is the approximated distance in here. Since the snake can cross from this side to that side without any stop, without any obstacle, it means the approximated distance is 0 because the snake can cross through this entire path without any obstacle. Is 0 less than edit distance threshold? As you can see from this maze size, we have about 5 rows. So it means we choose the edit distance threshold to be 2 because we have 2, 2, and the main path in the maze. because we said the number of rows is normally 2e plus 1. So from this shape, you could infer the edit distance threshold that we use to apply snake algorithm. And we said here 0 is less than 2, yes, so it means those two sequences are similar to each other. And if so, now we wanted to have the exact number of edit distance and not approximated edit distance. So that is being said, we already now sure that those two subsequences match each other exactly. Likewise for the blue subsequences and the green subsequences. And when we do pre-alignment filtering, now we are also sure that the things between every two subsequences are also similar to the things between the every two subsequences from the reference genome. Since we know now those information and we use pre-alignment filtering to approximate those numbers, now we wanted to do full exact solution at large scale using dynamic programming to be 100% sure that the number of differences now they do not exceed the distance threshold that we have. And this is the dynamic programming table that we should build. And we got some examples on how is this useful. We know that now using edit distance, we could get different solution with the same edit distance, but we also could get different solution with different edit distance. As you can see here, this is edit distance 5, but here it's 4. All right. And we also take some examples on edit distance calculation using the traditional method or the brute force solution and dynamic programming. We also went through Needleman-Wunsch algorithm for sequence alignment. And also we got, for example, Hamming distance, which could be used to calculate the differences, which should be substitution only. But also your colleague from this course explain about Smith-Waterman. So you have now multiple methods to apply for sequence alignment, which is the last step in read mapping. All right, so I hope I give you just a brief overview on the things that we have been doing through this semester, starting with indexing or introduction to bioinformatics, and then introduction, seeding, indexing, seeding, pre-alignment filtering, and all sequence alignment. And with that, we cover all steps needed for read mapping. Now let's see some read mapping results in action. So what we will get out of read mapping physically. So we already assigned you two data at the beginning of the semester called dummy.fastq and dummy.fasta file. So those two files, if you open them using Notepad editor, for example, I have here one of the sequences from dummy fastq, and I'm trying to compare it with what we have in the dummy FASTA file. So in this, I'm using brackets, Notepad called brackets. And in brackets, there is a very nice feature. When you double click on something, try to tell you whether it exists in the file that you are opening or not. So I did double click on this sequence over here, and you can see where it ever matches exactly in the FASTA file. So now we are sure that this sequence from the FASTQ file exists at location 0 in the reference genome or the dummy.fasta, and it also exists in this location. It also exists in that location here, here, and there as well. And this is very useful for small scale. So when you have a file that is very small, it's very useful to do that and check manually whether this sequence exists in the file or not. But imagine that we have real data coming from human that hold a large number of sequences and very large reference genome. We said like human genome has like 3.2 billion characters in it. That is a huge workload. Okay, So that is manually how you could verify the two files and see whether it exists or not. But in practice, we would like to use a tool for that. And the tool I'm using in this example called Minimap2, Minimap2, which is a tool developed by Professor Hingley. And this tool uses the minimizer algorithm to do the seeding and indexing. So they use minimizer seeds in this tool. Now, the tool is very easy to use. So what you need to do is just go search in Google for Minimap2 GitHub. And then you will find a repository on GitHub called Minimap2. And you can see the author name is Hingley. And this is how you can verify that this is the right tool that you should use. I will show you some example in a few seconds on that. And this is the command line. After you download the tool from GitHub, there are a few instructions on how you install it, very easy instruction. So you can install it in your machine, and then you could run it. And the way we run it is that we change the directory to the folder where the tool is and where are the executable of the tool inside the folder. So we change the directory to there, the active directory, and then we run the tool this way. Those are some parameters. For example, this parameter means don't report the read sequences that do not exist in the reference genome. So this means do not report the read sequences that do not exist in the reference genome. So only report the read sequences that could be mapped to the reference genome. And we give it the two input files, one FASTA and one FASTQ. And what it gives you a thing that we call SAM format. So now we have FASTQ, we have FASTA, and we also have SAM format. What we have in the SAM format, we have two parts, very important parts. The first one, we call it a header, and the header normally has things starting with @ sign. So those we call them tags, and all of those we call it the header of the file, okay? And the second part of this file is the actual mapping result. So this is one line. Let me maybe annotate it. So this entire thing is one line, and this entire thing is another line, okay? And this is, we said, the first part of the file, and this is the second part fully of the SAM file, okay? This is the header, right? This everything here, we call it header. And this, we have several lines in the SAM file. And each line, we call it a mapping result. Now let's see in a closed look what those things mean. So as we said, this is the whole thing is entire one line. And you can see in the line, there are columns. This is first column, for example. This is second column. This is third column. This fourth column, fifth column, sixth column, seventh column, eighth column, now ninth column. All of this is ninth column, and then 10th column, and so on. So those, everything is here. You can see a column and spaced by tab, another column, tab, column, tab sign, column, and so on. Okay. Now, what does the first column mean? This one. If you check the FASTQ file, the FASTA file, and see what's inside them, you will notice that this is the name of the read sequence. One of the read sequences, that exist in the dummy FASTQ file. So if you go back, let's quickly go back on here. You can see this name is exactly the name that we have in here, which is the first read sequence in the FASTQ file, right? And this is the exactly name we have it here. And then you will have some more columns. And now let's see the meaning of each of those columns. So this is a screenshot of one of the pages of the manual used for the SAM format. SAM format. And SAM format stands for sequence alignment, format, where we report the results of mapping a read or aligning a reads to a reference genome. So you can see in the first column, we always have the query name. The query name. The query name is the read sequence that we use to query the reference genome or to query the hash table. And then the second column is the flag. Flag. What does mean the flag? If you go to the next slide, you will see this very nice visualization tool for only the flag column of the SAM file. What we give here, we give a number, and this number in decimal, right? And then we ask the tool to explain what does this number mean. Now, this number is written as a decimal in the SAM file, but in fact, the interpretation of that number is not decimal, it's binary. So we need to convert the number, the decimal, into binary, and then every bit in that number has a meaning. Every bit in that number has a meaning. And those meaning are those things over here. If it is highlighted or tagged, if it is tagged, it means that bit is 1. If it is not chosen, as in those choices, it means those bits are 0. So we have 0, 0, 0, 0, 1, 0, 0, 0, 0, 0, 0, 0, and so on. And if you try to convert this number from 0, 0, 0, 1, 0, 0, 0 into decimal, you'll see that it belongs to 16. So 16 has the meaning of 0, 0, 0, 0, 1, which means read reverse strand. Let's skip the meaning of each of those, but we will visit the reverse strand toward the end of the slides. Okay, so every bit in the flag column has a meaning. And if you wanted to see the meaning of that column, you directly go to this link, to this website over here in the footer. And then you'll see something very similar to this. You just insert the flag number from that column in decimal and click on explain. It will tell you everything about that meaning. All right. And then we will have the third column. The third column in the SAM file means the reference name. The reference name. If you go back here, you will see the reference name is just highlighted over there. So this is the reference name. This is the reference name. What does it mean, reference name? If you go back to the reference genome we have, the FASTA file, you will see that the name over here is the name of the sequence. And this is another sequence that we have it in the FASTA file. So that name will be recorded in the SAM format if this read exists in the reference genome. If the read exists in the reference genome, you will see the name of the read, the first column, some flags in the second column, and the name of the reference genome that we are matching to as the third column, right? Now let's delete the annotation and move forward. Okay, so now what is very important is this column, column number 4, because it tells us the position or the mapping position. What does it mean? It means where exactly in reference genome I can start seeing the subsequence that is similar to the read sequence that I have. What does that mean? So when I record in the SAM file, I say that this read sequence is mappable or can be mapped this reference genome at which location? It looks like at location 1. So at location 1 in the reference genome of this read exists over there, right? And this is the thing that we already see using the brackets notepad, as you can see here. So this full sequence exists at location 1 in the reference genome. Again, you can start the location from 0. You could start it from 1. When we talk about computer science, it should start from 0 because it makes sense, because 0 is a storage, is a location in the memory. But when we talk about editor, which is general purpose and targets the public audience, we cannot consider the first character to be starting at 0, right? Okay. And then the fifth column we have is the mapping quality. Mapping quality. Mapping quality. What does that mean? It means how many matches basically we have in the between the read and the reference genome, and not only about the number of matches, but also do we have another mapping location in the reference genome that is also similar to what we have now. So if there are many locations as good as the first location, then the quality will drop quickly because we are confused. We don't know which one to pick. But if there are single location, single unique location that is similar to the read sequence, then the quality is much higher. We have very high quality to consider over here. The number 6, the sixth column, is about cigar string. And what is cigar? If you remember the arrangement that we did when we learned the sequence alignment, where we have the character like this, A, C, T, for example, C, A. Then we have another sequence, A, G, C, C, A, for example. And then we do some vertical bars, vertical bar, no vertical bar here, no vertical bar, but we have vertical bar here, another vertical bar. And we agree that those are arrangement to show the similarity. Now, how we present this arrangement in the file, we present it as what we call cigar string. So cigar string is a way to represent the similarity, the matches, the mismatches between two sequences when we after performing sequence alignment. That is the definition of cigar. It's very important. If you had it as a question somewhere in a conference, in an exam, in a quiz, what is cigar string? Is a way to represent similarity or the differences between two sequences after performing sequence alignment. Now, what is in reality? What is cigar? Cigar is, for example, if you want to represent this, we said there are 1, 2 matches. So we write 2M, it means 2 matches. And then 1 substitution, another substitution. So 2 substitution, for example. And then 1 match, 2M. So that is the cigar string that we will have. And this represents the arrangement that we show over here. So cigar string is a string that has a number of operation. Number of operations could be 100, 200, 1,000, million, which is how many of that operation. And then the operation type itself. So we could have matches, insertion, deletion, and undefined, substitution, and so on. Okay. Sometimes we don't represent the substitution, we represent it as M, it means mismatch or match. But this is how we represent the arrangement we have over here as cigar string. So we have the number of that operation, the item for the operation, and then we repeat that. So we have 2 of matches, 2 of substitution, 2 of matches. We also show if there are matches, we can write the reference name of the mate. This is in paired end, but we don't have to go through these details now, so we can skip this column. What is important also is the read sequence. So here we copy the read sequence as is. So we copy the read sequence as is, and we have it over here. Okay. And here is the quality, the sequencing quality of that sequence. We copy, column number 10 and column number 11, we copied them from the FASTQ file that we already have. Let's see an example. If we go back, this file, you will see this ID, this read sequence, right? You'll see that has also the read itself, which is here as 10th column. And we have the sequencing quality as the 11th column. You can see BCC, FD, DFE, and so on. Okay, I hope everything is clear now. And we see the definition of these columns. And this is what SAM file looks like. And this is how we could interpret or unlock the flags that we have in the SAM format. All right, so now let's go into a different topic in read mapping, which are the challenges that we have in read mapping. So now, hopefully, we know what is FASTQ file, we know what is FASTA file, we know how to solve the puzzle and map the FASTQ into FASTA file, as we see in the example. So we do indexing, we build hash table, then we do seeding, we do pre-alignment filtering, and then we do sequence alignment. And only if at the last stage, where we have the sequence or the edit distance is below or equal edit distance threshold, then we report the output in a SAM file. Okay, and hopefully by now we already know what is a SAM file. So we have a header, we have a couple of lines, each line represents a mapping. So when we infer from the seeding, when we query the hash table and we got the location as a result, we know now that the read maps at location 0, for example, in the previous exam. And based on that location, we are going to create an entry in the SAM file if sequence alignment went well and the edit distance was below or equal to the edit distance threshold. And with that, we report it in the SAM file. I hope that is clear. Now, the challenges with read mapping are many because it's a very important step. We have different data. We have different sequencing technology that we are dealing with, but also we have a huge workload at the reference genome side. So the first challenge is the repetitive regions in the reference genome. So we have the reference genome. We have many pieces in the reference genome that appears exactly in different places in the same reference genome. And that's what we call a repetitive region. It means مناطق مكررة. معنى repetitive regions مناطق مكررة، يعني عندنا كذا منطقة في الريفرنس جينوم هم نفس بعض فقط تكرروا في الريفرنس جينوم، وبالتالي we call them repetitive region. طب ليش هي challenge؟ Why it's a challenge in first place? Because when a read comes from a repetitive region of the genome, it can be difficult to determine which copy of the repeat the read belongs to. يعني لو كان عندنا reference genome تمام وكان عندنا read. Now, if this reference genome has many repetitive regions, right, and this reads كان أصلا مأخوذ من واحدة من هذه المناطق الموجودة في الريفرنس جينوم، وبالتالي النتيجة حتكون إنه لما نعمل مابينج حنشوف تشابه بين الريد وبين الريفرنس جينوم في أكثر من مكان. يعني حتصير هاي المنطقة مشابهة للريد، المنطقة هاي كمان مشابهة للريد، المنطقة هاي مشابهة للريد، بيعتمد على عدد الريبيتيتيف ريجن والريد نفسها من وين كانت جاية. إذا أجت من واحدة من هذه المناطق اللي هي مكررة أصلا في الريفرنس جينوم، فحيصير عندنا مشكلة إنه السام فايل حينتلي أو يصير مليان من أسطر من المابينج لهاي المناطق فقط. وكمان حيكون صعب علينا نحدد إنه هاي الريد جاية من أي مكان بالضبط من الريفرنس جينوم بسبب التشابه العميق في الريفرنس جينوم. الآن هل التشابه هذا أو هل التكرار موجود في الريفرنس جينوم حقيقي؟ طب ده حقيقي، كم نسبته؟ هل نص الجينوم؟ هل ربعه؟ هل 10% منه فقط مكرر؟ طبعا هذا بيعتمد على نوع السبيشيز أو نوع الفصيل اللي بنحاول ندرسه. وبالنسبة للإنسان، 50% من الريفرنس جينوم تبعه هو عبارة عن تكرار. 50%، نسبة ضخمة جدا. 50%، 50% of the human genome is made up of repetitive DNA sequences. وبالتالي نص الريفرنس جينوم هو عبارة عن فقط تكرار. تمام. اوكي. واللي مهتم بهذا الموضوع عشان يعرف أكثر كيف وصلوا لنسبة 50% ممكن يقرأ البيبر هاي. هو عبارة عن ريفرنس ممتع جدا وانتم تقرأوه إن شاء الله. وهو لما أول ما اكتشفوا الريفرنس جينوم، أول نسخة من الريفرنس جينوم اكتشفوها في سنة 2001، في بداية 2000، في نهاية أو في نهاية 2000 في بداية 2001. اكتشفوا الريفرنس جينوم او استطاعوا انه يعملوا ريفرنس جينوم للهيومن بعد 13 سنه من الدراسه. وطبعا هذا كان جهد عالمي بين عده دول من العالم عشان يعملوا النسخه الاولى من الريفرنس جينوم، وهم اول ناس عملوا ريبورت انه في 50% من الريفرنس جينوم او الدي ان اي تبعنا هو عباره عن تكرار. وبالتالي إذا عملنا سيكونسينج لدي ان اي لهيومن لانسان، حنشوف سبحان الله انه نفس القطعه تكررت معنا في كذا مكان. وبالتالي لما نعمل مابينج نتوقع انه يكون عندنا دائما اكثر من لوكيشن متوفر في السام فايل لنفس الريد. وبالتالي حتلاحظ انه في كثير اسطر في السام فايل، اول عمود إلها هو نفس الاي دي تاع الاي دي تاع الريد، نفس الاي دي تاع الريد في العمود الاول موجود في عده مناطق في السام فايل. ليش؟ لانه الريد نفسها ظهرت في كذا مكان في الرفرنس جينوم، وبالتالي السطر الواحد فقط بيسع لمكان واحد انا اعمل له ريبورت في السام فايل. كل سطر في السام فايل هو عباره عن ريبورت لمابينج لوكيشن واحد. فاذا كان عندي الريد ظهرت في مكانين في الريفرنس جينوم بناء على الاندكسنج والسيدنج والبري الاينمنت فلترنج والسيكونس الاينمنت، اذا ظهر عندي مكانين وفيهم الايديت ديستانس اقل من الايديت ديستانس ثريشولد، فلازم اعمل ريبورت للمكانين في السام فايل. المكان الاول بعمله في سطر لحاله، بحط مثلا 16 عمود وكل عمود بعبي المعاني تبعته. مثلا العمود الاول بحط الاي دي تاع الريد، العمود الثاني الفلاجز، العمود الثالث اسم الريفرنس جينوم اللي انا عملت له ماتشنج، وهكذا وبكمل. بعدين بروح على اللوكيشن الثاني. طب انت اللوكيشن الثاني لنفس الريد، وبالتالي بحط الريد اي دي كمان مره في العمود الاول، بحط فلاجز، بحط اسم الريفرنس جينوم انت يا ريد وين كنت ماتشنج في اللوكيشن الثاني، بحط اسم الريفرنس جينوم. بعدين بحط كل الأعمدة اللي تعرفنا عليها في السام فايل. اوكي. فعندنا اول تحدي هو التكرار في الريفرنس جينوم او الريباريتيف ريجن. تمام. Now التحدي الثاني انه عندنا differences between reads and the reference genome. We have differences between reads and the reference genome. So reads can differ from the reference genome in a number of ways, including single nucleotide differences, insertions, deletions, and structural variation. عندنا المشكلة إنه لما نعمل sequence alignment ما بنتوقع فقط نشوف اختلافات بسيطة. احنا دائما نعتمد على الإيديت ديستانس ثريشولد كمعيار إنه هل الاختلافات هاي بسيطة أو كثيرة. إذا كان الإيديت ديستانس ثريشولد 50 أو 500 5000 and so on، فهو هذا المعيار اللي بنحكي من خلاله هل والله العدد مقبول أو غير مقبول من الاختلافات. ولكن المشكلة إنه الاختلافات أحيانا غير معدودة. كيف يعني؟ أحيانا بنسميها structural variation، يعني اختلافات في البنية بتاعت الدي ان اي. وبالتالي هاي الاختلافات مش حرف أو حرفين، لا هي دائما بتكون أكثر من 50 حرف. يعني إنه تشوف 50 حرف inserted، 50 حرف deleted. تمام. وبالتالي حيكون الموضوع صعب في السيكونس الاينمنت تشوف هاي. طبعا 50 او 100 او 200 او 1000 حرف انحذفوا ممكن تشوفهم في السيكونس الاينمنت، خصوصا اذا كان التو سيكونسز كبار كثير، يعني مليون مثلا هذا السيكونس ومليون سيكونس ثاني، وبتبني الدايناميك بروجرامينج تيبل وتحط الايديت ديستانس ثريشولد مثلا ل 2000، ساعتها ممكن تشوف gaps of size 1000. تمام. it's doable. لكن لما نتكلم عن مليون حرف تم حذفهم من الجينوم هنا بتصير المشاكل. ليش؟ لأنه الستراكشر فاريشن هو ضخم هو عبارة عن large structural variation larger than 50 characters، وبالتالي صعب نشوفهم كل مرة في sequence alignment، وخصوصا إذا الريد كان قصير، وبالتالي في عندنا دائما أسئلة بنسألها لحالنا: شو هو ال edit distance threshold that we should use؟ شو هو ال edit distance threshold value that we should use؟ Should we use a value of 5, 10, 50, 60, 70? Until now, I always ask you in the questions, in the quizzes, the homework, the exercises to assume an edit distance threshold of any value that you like, right? But normally what we use is we use a threshold of 5% of the read length. So if we have a read length of 100 character long, then the edit distance threshold, or the appropriate edit distance threshold, should be around 5 characters, okay? Because it's 5% of the read length. Now, in some technologies like nanopore, we have many sequencing errors, so it's not fair to use 5% as a descent threshold. So we should increase that number. And to increase that number, we should go up to 10% or 15%, depends on how many errors that sequencing technology can introduce. The second question we have is score penalty. So we went through some examples of score penalties, giving a penalty score to the insertion, deletions, substitutions, and so on. So we always ask ourselves what value I should use there, what score penalty I should use for matches, mismatches, insertions, deletions. So basically, what score penalty should we use for each edit operation? Which sequence alignment algorithm should we apply? Smith-Waterman, Needleman-Wunsch, edit distance, Hamming distance, and many more, right? So each of those algorithms has its own application, its own benefits, its own applicability, right? So we should be careful about it. For example, edit distance can calculate the edit distance and the type of errors and the arrangement of those errors. But when we have two solutions of the exact same edit distance, we cannot prefer one over the other because we always use a unit score, because the penalty for each edit operation for all of them is similar. All of them is 1. So I don't have preferences for one edit operation over the others. So that's why it's important to choose, for example, Needleman-Wunsch, because where we can specify the penalty for each edit operation, and then we can have some preferences for one arrangement over another arrangement, okay? In one of the videos that your colleague prepared last week is about a few different types of alignment. So what type of alignment should we use? Is it local, global, G-local, and so on? So local is Smith-Waterman, where we don't care about the first few characters and the last few characters in the sequences. So we want to do sequence alignment, but only for the middle portion of the two sequences, meaning that whatever we have as differences in the beginning of the sequence, we won't report it. So we just ignore it, ignore the first edit operation, and then we just start reporting the moment we have matches in the beginning and matches at the end. That's what we call local. So you can have a look at Smith-Waterman video that we have in this course. A global is the Needleman-Wunsch algorithm. So we already got some examples on it, some practical example where we fill the dynamic programming table for that. G-local is a mix between local and global. So G-local, either we worry about differences at the beginning or we worry about differences at the end of the sequence when we build the dynamic programming table. All right, so that is the second challenge. Now, another important challenge is that the strand bias. The strand bias, what does it mean? So if you remember from the first lecture when we studied the DNA, we said the DNA has the double helix structure. The double helix structure. What does that mean? It means that we have two strands here and there, right? And those two strands, if you look carefully at each of them, you'll see something like that. If you look carefully at the DNA, you will see at each of those DNA line or prop, we have those two strands. What are those two strands? So we have the first one, the upper one, we call it forward strand. So we can read it TGC, TGGA. And we have the reverse strand, where it starts from here. Okay, CAACTGG and so on. And this, we call it complementary strand or reverse. Why it's reverse? Because basically we try to do the complement to each of those characters, each character, right? So we read it from the right. So that's why it's reverse. So if you see the T is bound with the A, right? Because based on the chemical orbits in the the thymine has, I think, 2 hydrogen bonds between the T and A. That's why they bind together. But when we look at G and C, they have 3 hydrogen bonds, and then those could bind or be chemically stable together. So every C binds with G and vice versa, G and C. And every thymine binds with adenine and vice versa, like adenine binds with thymine. So whenever we have a T, we should have A in the other side. Whenever we have a G, we have a C in the other side, and so on. However, whenever we talk about reference genome, we talk about one of those sequences. We never take both of them, right? So either we sequence the first or the last. And then we said the reference genome has T, G, C, T, which are the characters coming from the first strand or the forward strand. And this is what we basically see in the reference genome or the FASTA file. So what if when we do sequencing, we got the reads from this strand? What if we got reads from that strand? What if we got some reads from this strand and some other reads from that strand in the same FASTQ file? If we try to do the mapping, if we try to do read mapping to all the time to the forward strand, try to assume this is the reference genome and try to do read mapping to there, we're going to lose this group of reads where we have some reads coming from the reverse strand or the other strand, which is the complementary. So in this case, we could solve this issue quickly inside the read mapping tool, because we already know in advance that if we have one of those two strands, either the forward or the reverse, we could assume the other one, because we already know that A binds with T and vice versa, C binds with G and vice versa. So if we have one of them, that is good enough for us to mimic this entire DNA 2 strands together. So how it is done in read mapping, for example. If we go to read mapping, if you go to sketch.c, which is the file that is doing indexing and seeding together in Minimap2, if you go to line number 109 in this file, in the GitHub of Minimap2, what you will see this little line over there that try to get a k-mer from, let me clear the annotation, try to get a k-mer from this strand and then assume another k-mer that's been calculated in the previous lines of 109 and try to get the k-mer from the other strand. So what it does, when we extract all the overlapping seeds, we say, what if that k-mer is coming from forward strand? So I'm going to pick 5 characters from the forward strand. And then I also assume, what if that k-mer is coming from the other strand? So I pick the 5 characters from the other strand. Although what I provide to Minimap2 is only one of them. So let's assume we provide only this guy as a reference gene, right? So what does Minimap2 do to solve this issue is try, when we have the reference genome, try to extract all overlapping seeds, right, as we did in the examples. Every time Minimap2 extract one of the seeds, for example, the first one, it make another copy of that seeds, but with the assumption that this is coming from the complementary strand. So basically, it's have this seed and have another version of that seed, this seed and another version, this seed and another version of it, this seed and another version. What does this version means? It means I go to this k-mer and I check what is the first character. If it is A, then I write in here, I write T. And then I go to the second character. If it is C, I write G over here. If this is, for example, G, and then I write C. If it is A, write T again. If it is T over here, I write A, and so on. And this is how I calculate the complement of this k-mer. However, because it is reversed, sometimes I could reverse those characters. So I could report this k-mer as T, G, C, T, A. So it will be reverse complement seed of this seed. So this is the blue seed that I extract from the provided reference genome as FASTA file. And this seed's the one I assume myself that I have also the reverse complement reference genome. What would be the seeds that I should extract? So in Minimap2, it does both of them. It assumes that I have the FASTA file over here, so it calculates all the blue seeds, but also it calculates all the red seeds as well. Okay, and then how we apply the minimizer algorithm? Should we apply the minimizer algorithm on the blue sets or the read sets or both of them? Minimap2 try to reduce that and try to do minimizer of each group of those. So it means if I have the blue, if I have the red set like this, and if I have the blue set, I hope the meaning of the blue and the red are clear. So the blue seeds are the seeds extracted from the reference genome that's provided as a FASTA file in Minimap2 tool. And the red are something imaginary. So we don't have it. So we don't have the red reference genome. It is just a reverse complement of the blue reference genome that was provided as a FASTA file. And then Minimap2 assumes that since we have this imaginary red genome, we also could extract the seeds from here. And then we compare every seed with its corresponding seeds from the other group. So we take the first from red and the first from blue and try to do lexicographical sorting. So we pick only the ones that is lexicographically minimum, exact same as we did in Minimizer. So we always compare, is the blue seed, the first blue seeds, lexicographically minimum than the red seed from the second reference genome or not? Okay, so as you can see here, if the one coming from the forward strand is less than the one coming from the reverse strand, and then Z, which is the choice of which one I should pick, then it will be 0. It means that I will consider only the blue seeds from here. And I do the same thing and to all the seeds. So I pick now the second seed from the blue and the second seed from red, and I decide which one to consider. And then I consider the third seed from the blue with the third seeds from the red. And I try to do the lexicographical sorting and see which one to choose out of the blue and the red groups. I also get the last blue with the last red and try to get the lexicographical sorting out of those two. And then I will have a heterogeneous group of bunch of blue and bunch of red as well. And those seeds the one will be considered for minimizer algorithm. So that is a slight change on what we explained, but this is things to solve strand bias problem. Strand bias problem means I don't want to bias the analysis to always say that this read sequence coming from the forward strand only, which are the things that provided as input as FASTA file to the read mapping. I don't want to do that. I always want to check the forward and the reverse complement strand and see if the read was really coming from this strand or the other strand without imposing any bias on the analysis itself. Okay, I hope this is clear. I hope the drawing is clear. I hope the meaning of the red group was clear compared to the blue. And I hope also the way we choose out of the blue as a representative and out of red as a representative was clear, where we compare the first seed from here and the first seed from there. Okay. Now, there are other challenges in read mapping, which are very challenging to solve. One of them is long read. Imagine that we have a large or small genome, reference genome, and we have a very long read. Imagine that nanopore technology can give you up to 2 million long or 1 million long read. Think about 10 million long, 100 million long read. That is crazy to be compared with a reference genome, because it's going to increase the number of seeds that you would collect during seeding step. So think about the read sequence. You try to collect all overlapping seeds. If the length of the genome is million, then we are going to collect million overlapping seeds, which is a huge workload to deal with. There are some other computational challenges like memory footprint. If you want to load the entire reference genome into the main memory to start the analyzing it, that will require huge memory to handle the analysis. Or how about loading a small portion of the reference genome one by one to the main memory, try to process it, and then move to the other one, the main memory process, and so on. Execution time is always a nightmare because we prefer to have very fast analysis, very fast tool to do everything for us. And if it is very slow, it's going to slow the analysis for everyone. Think about a hospital that has like thousands of patients where we want to do genome analysis to them. So we want to start with sequencing and then read mapping, where we try to guess what is the location of each of those reads, so to build back your reference genome. And then it's taking a week per patient. And then it's going to take forever to process all those large number of patients. Accuracy is another important aspect because I don't want to detect insertions, deletions that they do not exist. And so think about, we are using a very bad sequence alignment algorithm that is trying to predict or approximate edit operation that do not exist at all. That will be a problem because we are reporting that you may have this insertion in your genome, but it does not exist. That is really bad. We don't want such problems. And with that, we are coming toward the end of this lecture. But before ending it, I would like to give you some small hints on how we do that practically. So I would like to share the screen from the other device, so you could see practically how we do it. Yeah, I hope you can see the screen clearly. Now I'm opening the terminal on my machine. So what you can see is this black and white screen where I apply some commands. And now what I have in here in this window, I have Minimap2 tool installed already. I have Minimap2 tool installed already. How we installed Minimap2? maybe in a few seconds I can tell you the source to get the minimap too, but assume we already have it, right? And I would like to apply the command line that I give you in the slides, which is dot/minimap2, okay? And then -a, -x, short read, SR stand for short reads, and then this parameter called SAM hit only. It means just report the reads that map to the reference genome. If there are reads that do not exist in the reference genome that you are analyzing, don't report them. Now we will try to see with and without that parameter, see how is the output. And then what we do is provide the dummy data that I provided you in the beginning of the semester, which is dummy FASTA and dummy.fastq. Now let's see the output. This is exactly what you would get, which is an output of two parts. The first part is the header of the SAM file, and then the second part has the mapping location. So this is a read. Those two lines is single line. It's not two lines. It just, the number of columns is huge or large. That's why the line is wrapped into another line, okay? And likewise with this line over here, maybe I can make the font a bit, yeah, those two lines together, two lines together, two lines together. And you can see that it's telling you that the first read in the FASTQ file mapped to the reference genome over here at location 1. And also it maps the second read mapped at location 139. And also the third read maps at location 567. And also the fourth read in the FASTQ file mapped at location 1. Okay. Now someone could ask, based on the brackets notepad, we noticed that the first read, only this guy, it's mapped to maybe 6 locations in the reference genome. Why I'm getting only one of them over here? That is based on the parameter that we choose in short reads. So if we remove short reads, we might get multiple mapping location per read. So now we remove this short reads, and that's true. Now you could see now the number of mapping in the SAM file becomes all of this, not only 4 lines, but all of those lines are the mapping location. And you can see in the first column, all of those belongs just to the first reads. All those lines, we have 1, 2, 3, 4, 5. So now we have 5 mapping locations for the first read in the FASTQ file. And if you want to check the mapping location, you would see that it maps to 281, it maps to 1, it maps to 141, maps to 567, 421, and 4, and yeah, that's it until here. So all of those are the mapping location where the read maps to. And this is the cigar string, the cigar string. This cigar string over here, 70 matches, 70 matches. What does it mean? It means this read appears exactly in the reference genome. The read appears exactly in the reference genome, this one, with no differences at all, as you can see from the cigar string after performing sequence alignment, that there are no mismatches, no insertion, no deletion. But if you look at this cigar string over here, you will see that it's telling you there are 33 matches, 61 deletion, 29 matches, 1 deletion, and 8 matches. Likewise, over here, you can see there are a lot of variation, a lot of insertions, deletions, matches, mismatches, and so on. Okay. I hope this is clear on how we got the alignment results and how we use sequence alignment in real life, how we report them in what we call SAM format. Now, the second or the last part of the practical side after doing Minimap 2 is how we visualize the results. So assume I got a SAM file of thousands of lines or million of lines. I don't want to open the SAM file and try to check column by column and see by naked eye what every entry in the column is, right? That is very time consuming and very laborious. So I'm going to use a lot of effort, a lot of time just to try to make sense out of all those columns. So a preferred way to check the SAM file is to visualize. And there are many tools to visualize this. I'm going to show you one of the tools. Let me stop sharing here. Open the program. I have it over here. And then try to share the screen. Yeah. So this is a program called Tablet. Tablet is where we provide a SAM file and can visualize this SAM file to us. And I provide the, let's now, maybe let's do this. Yeah, let's do this together. It will be very helpful for you. Okay, let's go back to this. I would like to do everything we have. Try to do it from scratch, okay? All right, so now what I wanted to do, so now assuming I did the mapping, I don't want to do it with short read, so I just use -a minus SAM hit only. By the way, -a, it generates the cigar string. So if I don't use it, let's see, if I don't use it over here, so you will not see any cigar string in the result. Will not do sequence alignment, basically. So -a is to report the cigar string. So let's keep it. -a. Now you get the cigar string and the sequence alignment. Now I would like to save this result. So I will use this to save the output that you saw in the terminal to a file. I will call the file as dummy.sam. Dummy.sam, right? Now I save everything. As you can see here, this is the command that I applied. As you can see, the SAM file is not shown, is not displayed. Everything becomes inside that file. And if you want to see the file, you can do head and try to locate the file where we save it with SAM. Now you got the first few lines of the SAM where we have the header and few lines of mapping. Okay. All right. So now assume we got the SAM file. What we should do, how we can display it on tablet. Tablet is somehow demanding to do more, and not only to provide the SAM file, but also to use a tool called SAM tools. So what we need to do with the SAM tools, I already have it installed. So now let's go to SAM tool. Yeah, what I need to do with SAM tools is first, it's asking me to convert the SAM format into BAM format, from SAM to BAM. BAM stands for binary and binary alignment. Binary means compressed, so the file won't be readable. It's not like this one text format, but it won't be human readable. It will be compressed, okay? So how we convert it, I'm going to use SAM tools, okay? And then I'll use tool called view, and then you don't need to understand what is going on over here, so you can just copy it as is. I can give you the command lines. B -O, and then you provide the output name of the BAM file. Let's call it same name, but just change the extension into BAM. And then I'll give it the original file, which is .sam. Now, this command, oh, sorry, yeah. It's telling me the SAM file I provide does not exist because it's not located in here. So let's give it this path. It means change the folder into one level up. Now it works well. And now we can try to see the dummy file. .sam. This is the first few lines of the SAM file. Now try to see the BAM file. You will see it's really encrypted, something encrypted. You cannot read it, and that is the purpose of the BAM. The BAM is compressed format of SAM. So everything you will see is not human readable because there is compression happening there. That's why the size of the BAM is way less than the SAM format, okay? Now we did the first step for visualizing the file. So we convert the file from SAM into BAM using that command, using SAM tools. Now it also asked me to sort the file, because it makes it easier for them to visualize the reads based on the read ID. So they want to make sure, or based on the location in the reference genome, the mapping location. So if they want to see at location 1, what all the reads that map. Want to see at location 2 what all the reads that map at reference genome. Then at location 3 what all reads that map at that location. So it's easier for them to sort them based on the mapping location. And how it's that done? It's done based on another tool from SAM. So we'll use SAM tools, sort, and then we will specify the output with type BAM and the output name with dummy, but we will give it also the suffix of sorted, so to differentiate the file name. And then we will have the original file that we convert into BAM. But let's make sure this is coming in the same directory, okay? What does this command do is just get the BAM file, try to sort it, and produce the output or the sorted output into another file called dummy sorted or underscore sorted.bam. Now the last step is to index that file. So we just want to index the file. Indexing the file makes it faster to read. So we'll use SAM tools again. Now we'll use the tool from the SAM tools called index, and now we'll just index the BAM file, the sorted BAM file. Now we are done. We use those very simple commands to convert to BAM, to sort the BAM, and to index the BAM file. And that is ready now to visualize the sorted BAM file. So now let's open the tablet again. And now let me share my screen. Okay. Now let's see. Now I want to open the dummy sorted BAM again. Yes, I want to continue. And now what we got the read and where it mapped to the reference genome. So you can see that there is a read here, and this is the read ID, as you can see. This is the read ID, and tell you that this read is mapped at location 1 all the way to location 70. There is 70 matches over here, and you can zoom in and see even the some of the properties of those. You can see some of the characters that are reported in the SAM. Some of the characters are not reported in the SAM file, so it doesn't show them. Here you can see this is read number 3, read number 1 also aligned to location 400, 421 all the way to 490 with 70 matches exactly. But also this read, which is read number 3, aligned to the same thing, 420 to 490 with 7 matches. So you can zoom out, you can see the full picture. You can zoom in to certain region. If you're interested in a region that is caused by cancer, so we can zoom into that and try to see how the reads are mapped. You can also go to the other reference genome because the FASTA file has 2 sequences, one called dummy ref1, and we have dummy ref2. So you'll see a list of those references over here. And you can see again, there's a read map to the dummy ref2 called dummy 4. So this is read number 4, which is aligned to the second reference genome, not the first one. And it's visualized those things to you. So this is how we practically do it when we like to visualize read mapping results. I hope this was very useful. And as I promised, now I want to show you quickly. Yeah, I want to show you quickly how we could get Minimap2 and SAM tools installed. So now if you go to Google, you would search for Minimap2, you will see the GitHub, and this is the code for Hengli user ID. If you open it, you'll see the GitHub. You could download the code from here. Once you download it, you get a folder with everything inside it. And then you go inside the folder with the terminal. Then what you need to do is follow the guide on how you install it, where you just type simply make, and that would build everything. Yeah, so you just go inside the folder by using cd minimap2 and do make, and that will build the whole tool. We'll try to install it. Maybe we can do it together. Okay, so I already download the code from the GitHub. I unzipped it. I get a zip folder, so I do decompression to the folder, and those are the files that I get. This is everything I got inside the folder. So this is a fresh copy I just downloaded from GitHub. What I need to do just to make sure that I'm inside the folder, then try to do make, just type make, and that's it. Hit enter. When you type enter, you just start installing the tool, and that would be it to get the tool ready. And for the SAM tool, if you search SAM tools, you go to the website, you go to the download, then you would get a fresh copy from this, you download it, and then you try to install it using those instructions. Very easy. You could do it yourself. You go to the folder after you decompress the folder, try to build those, and then everything will work. You could also go to their GitHub if you want so, and you'll see SAM tools over here. You'll see some instruction on how you install it. And I use this basically, those steps to build. So if you type those steps, everything should work fine. There's also Conda version if you are in advance in those things, SAM tools. If you know Conda, you would directly install this version for SAM tools and should work perfectly without the need to get the source code to install it and then build the tool from source code. This is how I did myself, how I install Minimap2 and SAM tools. And I'm going to give you the command that we use in this lecture on how you run Minimap2, how you get the BAM format, how you compress from SAM to BAM, how you get sorting, and how you index the BAM. And for the last tool that we use called Tablet, not tablet for electronics, but tablet for SAM files. Yeah, this is the website. If you go here, you'll see download it, and then you choose your operating system, and then you'd use it directly as the one I'm using. So here you will see the download. There's another tool called IGV, but I find it annoying because it might not display the large gaps in the file correctly. But you could try it yourself. So you could use this tool and see. This is not the tool website. Download. Yeah, this is the tool website. This is another visualizer that you could use. You use both of them, but sometimes when the gap is very large in the SAM file, so the IGV cannot zoom out perfectly to see that gap in full. So I prefer tablets sometime over the IGV and so on. I hope this lecture was useful for you, where you can get some concepts on how we really use sequence alignment in reality, how we use the cigar string, how we use the read mapping results. And I hope that was useful for you to get introduced to one topic on bioinformatics. There are some other interesting topics, hopefully in different courses. We're going to learn about them, including assembly, metagenomics, cancer research, and many more. Thank you so much for everything during the semester, and hope to see you discussing some of the projects or some of the quizzes, homework, maybe before the final exam or after the final exam. And we will be discussing more about this topic, especially with your colleague that's preparing some about the short videos on different flavors in this course or different topics. So please watch them on time. Try to do your own homework as well, search for more resources. try to get introduced to different topics in bioinformatics, and we are happy to discuss them in later lectures or discussion session. الحمد لله على سلامتكم كمان مرة، وإن شاء الله تضلكم سالمين وبالتوفيق لكم. شكرا جزيلا، يعطيكم ألف عافية والسلام عليكم ورحمة الله وبركاته.